In this video, we're going to talk about the specifics of the design of an experiment. Remember from a previous video, we discussed the differences between an observational study and a designed experiment. When looking at an observational study, we can only claim that there is an association possibly between our explanatory and response variables. In other words, we cannot claim from an observational study that the explanatory variable causes the response variable. We have to do that only in the designed experiment. And in a design experiment, we're going to intentionally change something to see how that response variable reacts. So we're going to talk about some terminology here. The first term we want to look at is called an experimental unit or subject. And in other words, this is going to be the person, the object, or something else that we are applying that treatment to. Then we have a control group. Now, because we are dividing our experiment up into groups, the idea is you put people in groups, you put things in groups, and again, you intentionally change something from one group to the next. We generally have a control group. The control group serves as that baseline for us to be able to compare treatments to. Then we have a placebo. Most people are familiar with the term placebo or maybe placebo effect, and this is kind of what you think, right? The placebo would be the pretend or the fake stuff that you might encounter if you're changing something in an experiment. When you think about a placebo, you want to make sure that your placebo is going to have the exact same look, taste, and smell as what the other stuff that is being given so that, there's no bias or it doesn't give it away right away. Then we have what's called blinding. In blinding, in particular, a single blind experiment, the single blind is when the experimental unit or the subjects or those participating in the experiment do not know what treatment they are receiving, but the researcher knows what they are giving. Contrast that to a double-blind experiment. And in a double-blind experiment, neither the researcher nor those participants in the study or the experimental unit know what they are being given. Generally, this helps to try and alleviate any type of bias, unintentional bias, or anything like that that can happen in the design of our experiment. Now let's take a look at an example of an experiment and see if we can identify some of these characteristics from the experiment. In this example, researchers conducted a double-blind, placebo-controlled, repeated measures experiment to compare the effectiveness of a commercial caffeinated carbohydrate electrolyte sports drink with a non-caffeinated carbohydrate electrolyte sports drink and flavored water. So you can see right away that they have taken 3 groups and they have 3 different treatments for these groups. One is going to be a caffeinated electrolyte sports drink. The other is non-caffeinated. And then finally, you have your placebo flavored water. So the participants in the study, 16 highly trained cyclists, each completed 3 trials of prolonged cycling in a warm environment. One time they received the placebo, one they received the non-caffeinated sports drink, and the third they received the caffeinated drink. It goes on to tell us how they completed the trial and that the total work in kilojoules performed during 15 minutes was used to measure the performance. It also goes on to further explain that the beverage order for the individual subject, so right here, the beverage order for the individual subject was randomly assigned, meaning each time the cyclist came up, it was randomly generated which one of the 3 groups the cyclist was going to receive, and they ended up doing this. Each cyclist ended up receiving all 3 treatments, but they were randomly assigned which treatment they received for their particular trial. And then it goes on to say that they tried to make all of the environment the same every time the trial took place. In the final paragraph, you see it says that researchers found that cycling performance as assessed by the total work completed during the ride, was 23% greater for the caffeinated sports drink than the placebo, 15% greater for the caffeinated drink to the non-caffeinated drink. And when they compared the non-caffeinated drink to the placebo, there was not a significant difference. So they were comparing caffeinated to placebo, 23% greater for that return. Then they compared caffeinated to non-caffeinated, 15% greater return for that one. And then again, when they compared non-caffeinated to water, there wasn't enough of a difference. Researchers were able to conclude that the caffeinated sports drink substantially enhanced physical performance during prolonged exercise compared to the non-caffeinated and the placebo. So let's go back and identify a couple of things that we've defined on the previous slide. In the first sentence here, you can see that they are telling you how they conducted the experiment. They had a double-blind, placebo-controlled experiment. Remember, a double-blind experiment, we learned, is where neither the researcher nor the participants know which one of these three drinks they were receiving. And then the placebo-controlled was that flavored water, which allowed us to be able to compare caffeinated, non-caffeinated to not only each other, but also to just plain water. Then let's kind of identify the population. Remember, in an experiment, we want to identify the population that we can make a conclusion back onto. And in this particular case, the population is down here at the very bottom, it says researchers concluded that the caffeinated sports drink substantially enhanced the physical performance during prolonged exercise. So who are they talking about when they're talking about prolonged exercise? You might want to just kind of consider that as endurance athletes. So that would be kind of the population that they would be looking for this prolonged exercise. But who is the sample that they are taking? The sample comes from these 16 highly trained cyclists. So the 16 cyclists would be the sample that they are pulling, and then they are going to infer back on the larger population. Now, I kind of have a question about that particular concept, just as a side note. If we've learned before that when you sample, you're trying to be able to take information, as much information that's representative of your population, so that you can then infer it back onto them. And I would probably argue that if you're only sampling cyclists, that there are other endurance athletes out there that maybe should have been involved in this trial to be able to infer back on endurance athletes as a whole. But that's our population and our sample. Now, what were the treatments in this one? Remember, the treatments were the caffeinated and non-caffeinated drinks, right? This is what we are using as our explanatory variable to try and explain whether or not these drinks, the caffeinated as well as the non-caffeinated, are going to have any type of enhancement to prolonged exercise. That's our treatment. And the response variable, in other words, what did they use to measure whether or not it enhanced their performance? That comes here in the center, and it says total work in kilojoules. In other words, they were measuring this performance to be able to determine that, to be able to answer their question. So total work in kilojoules becomes a quantitative because it's a measurement that we're able to take. So the output variable is quantitative, and we might kind of go back and say the treatment variable or the explanatory variable, remember that's the sports drinks, that would be qualitative. So here you've got an example of those terms that we were discussing earlier and what that looks like in a particular experiment.